Imagine slowly forgetting the names of the people you love. Imagine your hands no longer doing what your mind tells them to do. This is the reality for millions of families living with Alzheimer's, Parkinson's disease, or amyotrophic lateral sclerosis. As our population ages, these diseases are expected to affect many more, placing an enormous emotional and financial burden on families and healthcare systems worldwide. And yet, today we can only slow symptoms. We cannot stop the disease. For decades, we believed these diseases were simple in one way. A brain cell accumulates toxic proteins, fails to clean itself properly, and eventually dies. A slow, tragic breakdown, one cell at a time. But what if something more active is happening? What if sick brain cells don't just die? What if they spread the disease? Instead of destroying toxic proteins, the neuron secretly packages them into tiny particles called exosomes, microscopic messengers, and releases them. It's like a dangerous version of hide-and-seek. The diseased cell hides the damage and makes the neighbor sick. I call this the hide-and-seek mechanism. To test this idea, we use high-resolution live cell imaging to watch neurons in real time. We can actually track these tiny exosomes as they move between cells. By combining patient-derived cells and gene editing tools, we can try to block this escape route and see whether the spread stops. This is high-risk science, but if it succeeds, it could transform how we fight brain diseases and give families something that they desperately need: time, hope, and maybe one day prevention. If we are right, this changes everything. Instead of treating symptoms, we could intervene early. We could identify new biomarkers, design targeted therapies, and potentially stop disease progression.